Best of luck, everyone.
Oh,
Okay, I think we're live.
Should I double check?
Hold on. Yeah, I do want to check Messenger. Okay. I sent you the link.
Yep.
Yeah, again, very low-tech solution.
We're trying. You never know what it is.
Is
I
it?
might have set it to 30 seconds today.
Oh, that's all right.
Yeah, so far the title for this podcast is Cambridge Biologist Tutoring Refugees and Working in Science Outreach. So hopefully we can come up with a better one during the... We saw that on your CV.
Yeah, I mean, I did that for like half a year. So.
Oh, here we go.
Okay, fantastic.
Right. Welcome back, everyone, to
Wait, can we restart that? Sorry, I was just setting up. Okay.
Right. Welcome back, everyone, to what I believe is our 5th episode of our podcast. Yes, I'm surprised too that we are still doing this. But yeah, welcome back to Young Changemakers Podcast, where we talk to young people who are doing really, really cool things. My name is Blue. I'm one of the co-hosts, and I study medicine. And
I'm Sagav. I'm the other co-host, and I study maths. And today we've got, I guess, a change in tone. We've had very physics and maths heavy people so far.
know.
And we've got a vastly more interesting person this week. Not to complain against anyone. Teresa, do you want to introduce yourself?
Yeah, sure. So I'm Teresa. I study biochemistry in Cambridge, and I'm originally from Germany. And yeah, I would say I'm mostly interested in the molecular process of biology and also synthetic biology, so how we can engineer biology to make something useful out of it. And also drug discovery. So yeah, not quite specialized yet, but yeah, I like many different areas in biochemistry.
I have to say, I'm so happy we finally have a non-physicist and non-mathematician, because the past 4 episodes have been entirely mathematicians and physicists, and my brain is just. But so Teresa, how did you become interested in biochemistry or pharmacology? What drew you to it?
Yeah, so I think one of the, most important experiences for me was in high school when I was in 8th grade. A guy came to our class and just presented a science program at the German Cancer Research Center. And it's a pretty cool program where you can join different science groups and learn lots of different things. You can work on science projects, you can do lab courses and that kind of thing. And I thought, oh, it sounds cool. I didn't really know what he was talking about, but I knew it sounded cool. So I was just thinking, okay, maybe I should sign up. And I applied. And then the next year, so 9th grade, I was in the program. And so yeah, I did that program for from 9th grade until I graduated from high school. And it was really cool because it literally connected high school students and 9th graders with university students that are really passionate about what they're studying. I mean, I think a lot more passionate than many teachers are, and that know what they're talking about, and they could actually show me what science is like. And I mean, when I was first starting there, I was in 9th grade. I didn't really know I knew that DNA existed, but I didn't really know what DNA is made out of. And I heard all of these things, and I was surrounded by also 12th graders and so on. So it was a bit overwhelming, but that kind of drew me in, and it excited me that this sounds really cool. And so that's how I kind of got connected also to synthetic biology in the first place, because I joined the synthetic biology group and learned what they were doing. And I think that experience, and then later on I did also do a project in that student lab, and that was also really cool. And I think especially this process of working on a project and developing it yourself, and then getting over all of these little problems, and then actually getting some results, I think is really exciting. And so that's, I think, what really motivates me. I mean, apart from that, my parents are also scientists. And they also do biology, so it is also very, very related. But I wouldn't I wouldn't say that they influenced me that much to do science. I think they actually discouraged me also in some ways to like see other things, which obviously didn't work, but that was what I tried to do. And my grandma actually told me a few weeks ago that apparently I said when I was, I don't know, like 5 years old, that I never want to become a biologist because I see how much my parents work. I don't remember saying that, but maybe I did say that. But obviously, like I heard also them talking about science at the dinner table and so on. And obviously now they're also happy that I do science and like we share articles and so on. But they didn't necessarily try to like push me into doing biology.
Yeah, that's really interesting. Yeah. So I was going to pick up on, you said when you started the project in 9th grade, it was all quite overwhelming and, all new. Is that a feeling that sort of goes away, or is that something you keep on encountering every time you start something new? Or...
I mean, definitely, you're always overwhelmed and you can never really know anything completely, I think. And that's also, I mean, you just have to like start structuring it in a way and start reading. You have to start somewhere. And so don't let yourself be discouraged because you think you don't understand it. And I think because it can be discouraging. I never really liked, these these toys that people give you to like learn about, I don't know, electric circuits or something like that. I never really liked them because none of them actually explained it. And I was like, okay, why does everybody else get it? I don't get it. It looks cool, but I don't get it. And if I don't get it, then I don't really find it cool. So yeah, you don't have to feel like you understand everything, and you shouldn't feel like everybody else around you understands it. And yeah, at some point you are going to understand a few things. And then you go to uni and then they tell you completely different things, and then they tell you that everything at school was anyways wrong. So yeah.
That just hits really close to home. So you were talking about all the science projects you did during high school.
Mm-hmm.
So can you tell us a bit more about what kind of things you actually did about your synthetic biology project? And also, I read that you actually help coordinate those projects now.
Yeah.
So can you also tell us a bit more about that?
Yeah, so there's quite a big competition in Germany, which apparently is like the biggest youth science competition in Europe. So I think around, I forgot the number, but like many thousands of students take part a year.
If you didn't pick up on that, that was a very subtle flex.
No, but like it's a very big competition. So like in every school they promote it. And so I heard about this already when I was in like 6th grade and so on. I thought like, okay, this sounds cool, but I don't have a project. I don't have an idea. I don't know what to do. So I never really followed up on that. But then, for example, in 10th grade, I was part of like a summer science academy thing. And I actually did like IT. And so they kind of told us what computer is composed of and like the little, like the logic gates and so on and everything. And then we kind of built we simulated a computer. Mm-hmm. And so by doing that, I just like learned how it basically works. And I thought, okay, maybe now I know something and I can start a project. So that was my first project for that competition. And it's maybe not the most scientific project, but I mean, I was in like 9th grade. So what I did is that I built a marble machine that works the exact same way as like the logic gates in a computer does. So you have 2 different size marbles for like the zeros and the ones in the computer. And then it was like a system that they would fall down or go in different directions. And then at the end, it could only calculate 3 plus 3, but like a maximum to 3 plus 3. But the theory worked, and that was my first project. But
honestly, same.
Yeah, I mean, yeah, as I said, you have to start somewhere. And then when I started with a synthetic biology project. And so the way that came about is because I was in the synthetic biology group, and then they wanted to like restart a project. And then so they were like asking for people who are interested, and then I applied to like start that project. And then and so at first it was like 8 students and 8 mentors or tutors, or however you call it. And at some point, because people were leaving, going off to university and so on, it was like 3 students and 5 mentors. But it was very much like we were all collaborating and all just gathering ideas, and it wasn't like they were teachers; they were also friends. So what we were actually doing is that we were interested in modulating the energy metabolism of bacteria, and we wanted to make more ATP, which is kind of like the currency of energy in cellular systems. And so we wanted to either make that, or we just wanted to make a lot less ATP, so having kind of like a switch that could tune it in both directions. And so we came up with a system, and in this system that we came up with, it was just a bit difficult to explain without drawing anything. We realized that we needed like a channel that can conduct protons across the membrane, and that we can activate with light. Problem is, there is no such channel for bacteria. They exist in other organisms. Also, you can use them for like optogenetics in the brain and so on. But like, it's not there yet for bacteria. We wanted to have it in bacteria to do useful things with that. So we thought, okay, maybe let's just build it ourselves. And so we took a proton pump, and we wanted to convert that to a proton channel. And so the way we did that is just we did some mutagenesis. So we changed the sequence of the protein that encodes that, and tried to see if that makes it into a channel. Of course, we didn't do that randomly, but we found like a research article that did something similar. And so we thought maybe that could work. It didn't really work at the end, but like the theory was that it would work. We also like then simulated everything, like a mathematical model and so on. And I mean, the mutagenesis and everything that worked, but like the whole system then together didn't completely work. But either way, it was a really cool experience, and I learned a lot. And yeah, I kind of did this project over like a 2-year period of, I mean, obviously not full time because I had school, but like on the weekends and so on. And that was really cool, because I think like if you ever have the chance of actually doing kind of like a project, it is really rewarding, because I think that's basically the process of science in any field. So you read lots of things. So it took us half a year to come up with the idea. Then it took us half a year to like read things and try to make sense of it, then contact different labs and ask them for help. And then it took us like almost a year to do the experiments in the lab. And obviously, if you do like a real, I don't know, PhD thesis or something, it's going to take even longer. But then, so yeah, it's a stepwise thing, and you learn so many things along the way. And then also, if you then present your work, which is like part of the competition and so on. So I think that was really, really valuable experience. Yeah.
That's so fascinating. Yeah, I think like that's definitely not a problem, but with like, you have like summer research projects. That's definitely something that's kind of not missing in a summer research project, but sometimes they're only like 8 weeks long, so you don't actually get to experience like the full process of setting up a like experiment or research. Whereas like if you're actually doing like a master's or a PhD or like during the college time research project, you might be able to experience that more.
Yeah, the whole process, not just so we have this idea, so you're going to do these experiments and you're going to get these which is why, I mean, obviously like some internships are great, but especially if you thinking about like labs that you would do for your course, I think they're really just not what you would do in an actual lab. I mean, you learn the techniques and it's useful, but I think some people think that that's what research is like, which in my experience so far, it just isn't. It's a lot more exciting when you actually don't know the outcome. So, but also, apart from that, I mean, obviously, like it was really great that I had these opportunities, and not everybody has them. But like even if you're just in high school, I think it's so valuable to like try to reach out to labs, because generally people do this because they're really fascinated and they want to find out more, and they want to share knowledge, and because they do quite idealistically, I would say, and sometimes they're very open to share this with you. So, for example, when I was in 11th grade, first of all, I really wanted to go to Stockholm, but also I really wanted to see a lab. So I thought maybe I could just combine these two things. And I just emailed like a ton of labs, and then eventually one got back to me. And he was like really excited that a high school student, and obviously I also like tried to make sure I understood kind of what the lab was doing and so on, was like interested in his research. And he even offered me to like stay at a certain place for a week and like have accommodation and everything. Which I mean doesn't happen anymore when you're at uni, right? Because everybody has to like find internships, and you're not kind of special anymore because everybody's an undergraduate student. But if you're a high school student, just go for it and just email people, because I think they'd like to see that you take the initiative as a high school student. And then not that many high school students do that, and then you end up getting a really interesting insight. I ended up being there for like a week and just like following different PhD students, and that was really cool. And I had really great conversations and discussions with them, and I can highly recommend that.
I kind of wish I heard this 4 years ago.
Yeah, but I think that's something we always try and talk about in our podcast, is like how important it is to cold email people, call, email people, because especially when you're in high school, because I think the moment you leave high school, it just gets harder and harder and harder. Exactly. And like even the moment you graduate uni, it's just like people are just like
competitive, and many people want to do these things. So if you're just a little bit earlier and just do it 5% differently, then you can see so many things. I think that helps a lot.
And like there's no harm in emailing. You know, like the worst it's going to do is they're going to put you in.
I can tell you for that Stockholm internship, I think I emailed 9 people. So it felt a bit disheartening. I mean, but then most people got back to me and were like, "Oh, we just don't offer for high school students," and so
No, I can definitely relate. I had to get work experience to apply for medicine, and I emailed like every single hospital in London, and one got back to me.
Exactly. Yeah.
But it was like it felt so good when they were like, "Yes, we'll take you on," and like exactly. And they were like cystic fibrosis, because it was at the Bromptons, so that was all like cystic fibrosis, primary ciliary dyskinesia, all of that. And I'm still doing cystic fibrosis research now, so it's kind of cool how like I don't know, what's the word, synergy or something. So Teresa, you also you help coordinate the projects there now as well.
Oh yeah. I mean, because
so really cool.
And then apart from the project I told you about, the synthetic biology one, I also did a chemistry project at school at the same time, also with Jenny, by the way.
She's on our podcast.
Surprise!
Oh really? Okay. Anyways, I really like doing these projects, and so I thought. So actually, a friend who was also in the synthetic biology project initially, she also did like a lot of projects for this competition, and she was like, "Okay, do you want to like start like helping students in this science program do like these kind of projects and help them prepare for the competition and so on?" Because the other people that organized that before they kind of left, and then so we just started doing that, and I think it's fun. At the moment, we have around 6 teams that are going to participate next year, and then also like new people coming in. So what we try to do is just like find mentors or supervisors for them, get money for them, then like set deadlines for them, read their summaries and like reports and everything, because that's part of the competition. And it's really interesting, and because I mean I get to experience all of these projects while I'm actually not doing them. So that's actually really cool that I just get to see all these different things, and I can kind of share my experience and
you're like giving back to like what
getting excited about their project is really cool.
That's great.
Since we're already talking so much about research experience, do you want to tell us a bit what you did at the ramen lab, I believe, recently?
Yeah. So, I mean, you all noticed the pandemic.
The what?
Pandemic also.
What rock are we living under?
No, I mean the pandemic also had also shifted my summer plans. I was planning on doing an internship in London at the Institute of Cancer Research, and so I had everything. It was probably a few months, and then it got cancelled. And at the same time, our exams basically got cancelled, and so there was just nothing to do. And I thought I want to do something because studying for exams that don't really count didn't really motivate me. So I just contacted a supervisor, or not a supervisor, a lecturer from my pharmacology course that I did last year. And I was just saying, yeah, I was interested in your lectures because of XYZ, and I would like to learn more. Could you just give me a project? And so, yeah, he was also excited about that. And then he was like, okay, sure, we have COVID, so why don't you start working on COVID? So his lab usually doesn't work on coronaviruses, but the lab generally does a lot of modeling. So kind of trying to model how small molecules would bind to proteins, and like which proteins would bind in which way, and in which on which side on the proteins, and so on. And so he thought, okay, you could just take like one of the key proteins in the virus, the main protease, which is responsible for kind of chopping up the proteins that are made into functional proteins. And I could just take that and try to see if I can find something that would bind there. So yeah, he actually like showed me how to use the programs and so on, but then he just said, you can do whatever you want with it, which was really cool because that's what I did. I just started looking for compounds that were active, for example, in like the previous SARS coronavirus that caused the epidemic in 2002, and because they're very similar. And then I just tried like docking them, so simulating how they would dock to that new protein. And I also started looking at like drugs that are currently in use and repurposing them, which actually a lot of people did and are doing now. And I did find some interesting things, but then obviously because everybody's looking at coronavirus at the moment, the things that I found were also other people's findings, and so I couldn't really share anything new. But he was like, okay, sure. So now you know how the programs work. Why don't we do another project? And I was like, yeah, my summer internship got cancelled. Why not? So I started on a new project, and this time it wasn't just me; it was also with a PhD student, which was really great because we could collaborate and share thoughts. I also did that with the supervisor, but I mean he's a group leader, so I didn't want to bother him with my emails all the time. Exactly. And then so we started on a new project, and this one was a little different than I guess most of the studies that are now published about coronavirus, in the sense that we used some of the data that was produced from a COVID-19 moonshot initiative that did a lot of, they used a lot of fragments, and then they crystallized it with the main protease. So generally protein crystallization and then structure determination is a very essential step and big step in structural biology, and it's really important. And they did that. And with these fragments, you can of course build bigger compounds that could act as actual drugs. Mm-hmm. And so we used the fragments, and we just looked in a database for compounds that are kind of similar to that in their pharmacophore features. And then we just made a huge library of compounds that we would test, and then we did just the several steps of the simulation that I did before, and then kind of analyzed all of the outcomes and identified a set of some new scaffolds that could be used to then develop drugs for the main protease. Obviously, everything of that is just modeling based. So everything has to still be tested in experiments, actual experiments. But maybe there could be something that actually works on the main protease. That's the hope. So that's the stage where we're at the moment. And then soon we also want to like test these in actual experiments if it works. And yeah, I'm excited how that's going to work out. But yeah, it was definitely like a great experience to also see the more bioinformatics side of things, and which is generally very valuable to see many different kinds of ways you can approach the same question. Because I mean, a lot of things that other people do, they could also just as well do all of these modeling steps, and they would save some time. So yeah, I think it's a very, very good way to also see different areas that are kind of connected.
Yeah.
What do you think? So since COVID-19 has spread, there's been so many like research projects, well, like just research. A lot of labs who normally wouldn't be doing COVID-19 research, started doing research into this, and a lot of like trials and things have been like accelerated. What do you think are like the implications of all this? Like,
I mean, there are a lot of positive things coming out of it, such as like these initiatives that also like the initiative, it was completely, I mean, not completely, but they shared a lot of data without it being published. And without this data being available, we wouldn't have been able to do our research, which I think is great. And also just people, I don't know, dropping everything that they're working on and then jumping in on something else that, I mean, it's related and they can use their knowledge to do something good. I think it's really good. At the same time, obviously, some like important like studies and clinical trials and so on, they were put on pause that are also really important. So yeah, that's kind of the trade-off. And I also heard of quite dramatic things that people, I mean, obviously people that do experiments and work with cells and animals and so on, those experiments you plan over years. So it's quite difficult to like just disrupt it for a few months of lockdown. But yeah, I mean, I hope that everything gets back to normal eventually. But yeah, I think everybody's still in shock.
Definitely.
Clearly.
I think definitely with the pandemic, do you know previously with like the Ebola outbreak? I think it was Ebola, I might be wrong, but there for like one of the first times ever, there was a real like sense of like open source, like this whole idea of open source science, and there was like researchers from all over the world like actually collaborating, because I guess like science can be very competitive sometimes. Like everyone wants to publish the first paper or find the first protein or whatever. And I think it was Ebola. I might be wrong. It was another pandemic anyway. But they were talking about how, and I went to this talk there, it was like a time that people were really collaborating because they just really needed to find a cure very quickly to be able to like stop the outbreak from happening. And hopefully, like with COVID, I feel like it's kind of the same thing. We haven't quite found, well, we might have found the vaccine. No one really knows. But maybe it'll lead to like more open source collaboration.
Yeah. And I think, I mean, it's also a good thing that we become proficient at having virtual meetings and everything.
No, it's interesting. Yeah, it's interesting to see how like science is also changing. I guess I feel like technological advances like are very much accelerated. Like in history, they've always like accelerated in conjunction with like big historical moments, I guess. Then a lot of advancements came from like World War, between the World Wars. So I don't know. What do you think, Megan? Any mathematical advancements?
I mean, a lot of epidemiology has improved recently. I mean, I wouldn't say that the coronavirus specifically has had any impact on anything that isn't directly modeling biological spread within maths. But right, so I think, again, I hate being the person to always say this, but we should start wrapping up. So I guess, Teresa, what do you think is the most interesting thing in your field in the next 5 to 50 years?
Ooh, massive, but I think like generally the new technologies that are shaping biochemistry, such as that we're able to do like multi-omics. So we can look at the proteins at the same time. We can look at the sequences of DNA and combine all of that data and make sense of it. I think that's going to be hugely important. And then also, I mean, structural biology is also having like massive advancements with. So I mean, historically, it was mostly just like X-ray crystallography that was able to get high resolution, atomic resolution structures. And now, I mean, for already a few years, electron advancing to like also deliver atomic resolution structures, and and in a way more natural environment. And so I think that's definitely also going to contribute a lot. And I mean, obviously I'm interested in like synthetic biology and so on. I think like a lot of interesting ways of design nature have been like pioneered, or ideas have been about that, but then maybe not that many things have been applied necessarily. So I think then a lot more things that we've been like reading over like the last 20 years or 10 years and so on, they're actually going to have impact. So I think they're going to be developments on all fronts of like more applied and more on the basic science.
It's an exciting time.
Yeah, it is. And biology is awesome. I hope not. I can convince the people.
No, definitely. Thank you so much for coming on our podcast. I'm so happy that we had a biologist come on.
I mean, all sciences are interesting, but biology is just the most fascinating, because I mean, it's crazy that you can have so many different reactions going on in one cell, and then all of a sudden everything just like works together, and you can have complete organisms, and they can like feel and think and reproduce and everything, and then you can have proteins that work like motors, but they're not actually like designed by people. At least I would say it's not designed by people, but and they can just do that because they evolved that way, and then we can make them even better in the lab. And I think that's really cool.
Incredible.
Right. Thank you so much.
Thank you for coming on.
Yeah.